Hello students, and welcome to the third video of the immunology classes that I have been taking. So today we are going to discuss about the organs of the immune system. So as you know, the immune system is nothing but the defense system, which mainly comprises of cells, which we have discussed previously, the vessel system or the lymphatic vessel systems, and the organs. So today we are going to talk about the organs of the immune system. So the immune system is made up of organs which control the production and the maturation of defense cells known as the lymphocytes. Okay, so there are three types. Before that we will discuss about the organs. So many morphologically and functionally diverse organs and tissues have various functions in the development of the immune responses, and they can be differentiated into the primary, the secondary, and the tertiary lymphoid organs. So the primary lymphoid organs are known as thymus and bone marrow. Okay, and remember these names: thymus for the T cells, bone marrow for the B cells. Since the thymus starts with the letter T and bone marrow starts with the letter B, it's easy to remember. So these are the organs, or the primary lymphoid organs are those organs where the maturation of lymphocytes takes place. And the secondary lymphoid organs are examples are the spleen and the lymph nodes. Also the cutaneous lymphoid and the galt. Okay, the malt and the galt, which is the gut associated lymphoid organs, and the malt is the mucosal associated lymphoid tissues. So these all form the secondary lymphoid organs. What is the role of the secondary lymphoid organs? The role is to trap antigens and provide sites to mature the lymphocytes, where these mature lymphocytes can react with the antigen which is trapped. Now, there are tertiary lymphoid organs which normally contain very few lymphoid cells as compared to the primary and the secondary lymphoid organs. And these are normally into account they're taken when there is an inflammation or an inflammatory response. The tertiary lymphoid organs, also known as a TLO, are abnormal lymph node like structures that are usually seen in the chronic inflammation, and when they are summoned when there is a transplantation, undergoing a graft rejection, during some cancers, or during some autoimmune responses. Okay, so we will talk now about the primary lymphoid organs, which is the bone marrow and the thymus. So let's talk about the thymus. The thymus was first founded in 1961 by Miller, who was investigating leukemia in mice models. Okay, the thymus is an organ which is, I can show you. Okay, it's a bilobed organ, which is a flat bilobed organ situated above the heart, and each lobe is separated from each other by strands of connective tissue known as the trabeculae. Okay, and they are divided into two compartments. The outer compartment is known as the cortex, which is packed with T cells known as thymocytes. Okay, and the inner compartment comprises of the medulla, which is very less populated with the thymocytes. Both the cortex and the medulla are crisscrossed by a three-dimensional stromal network composed of epithelial cell, dendritic cell, and macrophages. Okay, so this is the structure of the this is the cartoon structure of the thymus. So you can see the outer and the inner structures that we have just discussed. Okay, the cortex and the medulla, as I said, you can see that they're crisscrossed with three-dimensional stromal network, and you can see the epithelial cells. Okay, I'll zoom. This is the epithelial cells, the dendritic cells, okay, interdigitating dendritic cells, as well as the macrophages. The macrophages are also present in yellow here. So this is the complex system of the thymus. So the outer cortex you can see, and the inner comprises of the medulla. So thymus has around they have many cells, also have nerve cells and long membrane extensions, and these have a lot of amounts of thymocytes, which is nothing but the T cells, forming large multicellular complexes. So they have the function of the thymus. What is the function? It is to generate and select a lot of T cells that protects the body from infection. As the thymocytes develop, the enormous diversity of T cell receptors is generated by a random process, and the T cells only work when they are presented with an antigen presentation complex known as the MHC molecules. And most of these T cells do not they are not capable of recognizing the antigen which is presented, and they have to be eliminated. So this happens right here in the thymus. The thymus induces the death of those T cells that cannot recognize the antigen MHC bound complexes. Okay, and those also reject those which bind to self antigen and causes autoimmune disorder. Most of, or more than 95% of all the thymocytes die out of apoptosis, which is a cell suicide mechanism, and they do not reach maturity, and only the functional thymocytes, which work in providing the appropriate immune response, will work and will be reaching maturity. The role of the thymus was studied, as I said, by Miller in mice models, where he removed the thymus, which is known as thymus ectomy or thymectomy, where the mice is then known as a nude mice because the fur is all lost. Okay, the mice is furless, and when this organ was removed in mice, the mice was susceptible to several infections and failed to reject the skin grafts, and also the mice died prematurely. So this was an observation that the thymus produced far more immune functions, and therefore the T cells or the thymocytes was named after the thymus, and this was found by Miller, as I said. So yes, we said that if the thymus is malfunctioned, so many diseases have been caused. So other diseases, they're like DiGeorge syndrome. So many kinds of diseases also take place when the thymus is absent. Okay, aging is accompanied by the decline in the thymic function. Okay, so the average weight of the thymus is around 70 grams in an infant and only 3 grams in a elderly person. So you can see the degeneration of the thymus. Moving on to the next primary lymphoid organ, we'll talk about the bone marrow. So the bone marrow is present in humans and mice, also in birds, but in birds they are it's a different lymphoid tissue. It's known as bursa of fabricus. So here what happens in the thymus, like the T cells mature in the thymus, in the bone marrow the B cells mature. So these B cells arise from the progenitor B cells, which proliferate and differentiate within the bone marrow, and the stromal cells within the bone marrow interact with the B cells, secrete cytokines that are required for development. So here also there are selections which eliminate the non-functional or the self-reactive antibodies. Bone marrow is not the site of B cell, as I said, in all animals or in all organisms, like birds have bursa of fabricus. So this is a very beautiful diagram, as this is a femur bone. Okay, you can see this bone is hollow from inside, but the spongy part of the bone contains the marrow. The marrow is nothing but densely packed cells. What are those cells? You can see they are the red blood cells, white blood cells, platelets. Okay, so this is how the bone marrow looks. So multiple bones support the bone marrow system, such as the hip bones, the ilium, the femur, the sternum, and the humerus. Okay, the lymphoid organs, they the B lymphocytes or the precursors of the antibodies are formed, matured in the bone marrow. So it is composed of hematopoietic stem cells, adipose tissue, and supportive stromal cells, and it is in adults. I told you all the places where it's located. The bone marrow transplants can be conducted to treat the diseases in severe conditions, and even sometimes during cancer such as leukemia, where the immune system does not function well. Okay, Now we spoke about the primary lymphoid organs. Let's talk about the lymphatic system. What is a lymphatic system? The lymphoid organs are connected to each other as well as to the site of the infection via circulatory systems such as blood and the lymphatic system. Okay, the lymphatic system comprises of several lymphatic vessels, lymphatic nodes. So we'll talk about them. The blood circulates under pressure, okay, and it's the fluid component or the plasma seeps into the thin wall of the capillaries to the surrounding tissues. Much of the fluid called as interstitial fluid returns to the blood via capillary membranes, and the remainder of this is now called as lymph flows back into the spaces in the connective tissue, and there is a tiny network, and they open up through the lymphatic capillaries into series of larger collecting vessels known as lymphatic vessels. This is a flow or the direction of how this network works. Okay, in this way the lymphatic system will capture all the fluid that is lost from the blood and return it back into the blood. Therefore it maintains the steadiness of the blood as well. The flow of lymph is achieved by the lymphatic vessels, and it is usually by the movement of the muscles. A series of one-way valve along with the lymphatic vessels ensures that the lymph flows unidirectionally, and that too only in one direction. And this is the intricate network. As you can see, this is the tissue spaces, this is the lymphatic vessels, the capillaries, these are the lymphatic nodes. Okay, this is the lymphatic node, the lymphatic follicle. So this is how it looks. Okay, so now what does the lymphatic system, why is it important? Because when the foreign antigen gains entrance inside the tissue, it's picked up from the lymphatic system like at the site of entry, and it drains all the tissues of the body, and it's carried to various organized lymphoid tissues such as lymph nodes, which then traps the foreign antigen. As the lymph passes away from the tissue to the lymphatic vessel, it becomes enriched in the lymphocytes. So it's like a passageway for the immune cells, so that the immune cells can trap the antigens and carry it to the or direct it to the organs or the different kinds of cells which require immediate attention. Thus the lymphatic system serves as a mean of transporting lymphocyte and antigens from the connective tissue to organized lymphoid tissues, where the lymphocytes may interact with the trapped antigen and undergo the activation. Okay, now we'll talk about the secondary lymphoid organs. The secondary lymphoid organs, they comprise of structures called as the lymphoid follicles, okay, which consist of aggregates of lymphoid and non-lymphoid cells surrounded by network of lymphoid capillaries. Only when it is activated, the lymphoid follicle is known as, which is the primary lymphoid follicle, will make the network of follicular dendritic cell and B cells. And once the antigen is exposed, or antigenic challenge is what we know it as, the primary lymphoid organ starts enlarging, grows in size, and becomes a secondary lymphoid follicle, which has a ring of round concentric arrangement of B lymphocytes, which has a center germinal layer in which the proliferating B lymphocytes are present, and they are also having the helper T cells and is mixed with the macrophages and the dendritic cells. So all this complex network helps in taking care of the antigen. Okay, most of the antigen activated B cells divide and differentiate into antibody producing plasma cells in the lymphoid follicles, but only few B cells in this activated population will reach the germinal centers. After the division and maturation, there is a rigorous selection process. Only the functional and the non-self attacking the cells, only they will remain, and most of the others will die out of apoptosis. In general, those B cells producing antibodies that bind the antigen more strongly have much more chances of survival than the others. The small number of B cells that survive the germinal centers, rigorous selection process differentiate into the two types of B cells, as you know, that is the plasma or the memory B cells. Okay. Now this is the lymph node. This is the typical structure, the outer cortex, inner medulla, and between the medulla and the cortex is the paracortex. What are lymph nodes? Lymph nodes are the sites where the immune response mounted to an antigen in the lymph. So they are the place where antigens are trapped. Okay, they are bean-shaped structures consisting of a reticular network packed with lymphocytes, macrophages, and dendritic cells. They are clustered at junctions of the lymphatic vessels, lymphatic nodes, and are first organized they are the first organized lymphoid structure to encounter and trap the antigen into the tissue spaces. As the lymph, the fluid, percolates through the node, any antigen that is brought with it will be trapped over here, and there's a network of phagocytes and dendritic cells. So the overall structure of this lymph node supports the ideal microenvironment for the lymph node to act in a particular way and respond to that antigen. Morphologically, a lymph node can be divided into three regions, that is the cortex, paracortex, and the medulla, as I said. Okay. So the outer layers consist of macrophages and follicular dendritic cells arranged as a primary follicles, and after, as I said, after the challenge, these enlarge, become secondary follicles with the germining center. Okay, after the germinal center, we will discuss about the lymph coming from the tissues. They percolate inside, and the antigen is trapped over here, okay, by the lymph. So now we'll discuss the second example of the secondary lymphoid organ, which is the spleen. Spleen, you can see the structure of the spleen. It is a very important immune organ which is large, okay, and it is present near the left abdominal cavity. When the lymph node is lymph nodes are specialized in trapping antigens. Here the spleen does not trap antigen, okay, but the spleen has other important roles. So it is used for filtering the blood-borne antigens, and it therefore responds to systemic infections. So here the trapping is in a different way. The lymphatic vessels are not drained into the spleen. Instead, the blood-borne antigens and the lymphocytes are carried into the spleen through the splenic artery. Okay. So the experiments that have been conducted, they show that the more circulating lymphocytes pass daily through the spleen than to the lymph nodes, all the lymph nodes in the body combined. Thus, the spleen is surrounded by a capsule which is having a number of projections known as the trabeculae into the interior to form a compartmentalized structure which has the red pulp and the white pulp, which is separated by the marginal zone. The splenic pulp consists of a network of sinusoids. What are sinusoids? They are low pressure vascular channels that receive blood, okay, and they are also populated by macrophages and erythrocytes, which are the RBCs, and very few lymphocytes. Okay, here this is a site where the old defective red blood cells are destroyed and removed to make space for new ones. Many of the macrophages within the red pulp contain engulfed red blood cells or iron pigments from the degraded HB hemoglobin. The effects of splenectomy, which is a removal of the spleen, was also studied, and it was found that the spleen, when it is removed, depends totally on the age of the person at which the spleen is removed. In children, if the spleen is removed, then it increases the risk of harmful bacterial sepsis caused by dangerous bacteria such as Streptococcus pneumoniae, Neisseria meningitis, and Haemophilus influenzae. Whereas splenectomy in adults have less adverse effect, but the person will be prone to bacterial infection or bacteremia. Okay, now we'll discuss about the CALT, which is the cutaneous associated lymphoid tissue. Now cutaneous means, you should remember the word skin, okay, because the skin is the outermost largest organ, outermost layer, and the several tightly packed epithelial cells are present, and specialized cells such as keratinocytes are present. The dermis is present, which is a thick layer composed of connective tissue which is in contact with the blood vessels, hair follicles, sebaceous glands, and the sweat glands. Now these sebaceous and sweat glands are important here. The sebaceous gland contains sebum, which is like an oily fluid that has acidic environment which inhibits the growth of most of the microbes, and the sweat on the other hand consists of the substance like lysozyme, which is known for its antibacterial activity. The keratinocyte and the skin mast cells secrete cytokines. Cytokines are small protein chemicals which are messengers that regulate the immune response. Okay, and the keratinocytes can be induced to express class 2 MHC molecules, which is, I told you, an antigen presenting molecule and therefore function as an antigen presenting cell. The skin or the dermis contains CD4 and CD8 T cells, which we will discuss later, for which helps the immune system to function better, and also they have the macrophages. The last immune organ which we will be discussing today is the mucosal associated lymphoid tissue, or the MALT. The MALT or the MALT is a diffuse system of small concentrations of lymphoid tissue found in various sites of the body, such as the GI tract, the thyroid, lung, salivary glands, the eye, and the skin. Okay, all these places are sensitive. The eyes, okay, the GI tract. So this immune response is populated by T cells, which are well suited to encounter antigens that enter through the intestinal mucosal epithelium. They contain the B plasma cells, the B cells, the activated T helper cells, and the macrophages in loose clusters. MALT plays an important role in regulating the mucosal immunity, and its main function is to support the immune system by trapping antigens and presenting it to the lymphocytes. Okay, so we have seen the primary cells, the primary organs such as the bone marrow and the thymus, the secondary immune organs such as the spleen, the lymph nodes, the MALT, the GALT. We have talked about the lymphatic structure, the lymphatic network, and one thing I forgot to tell you is about the Hassel's corpuscles, which is in the thymus. Okay, let me go quickly to the slide. Okay, so it's here. So what do these Hassel's corpuscles do? They are known as the thymic corpuscles, and they are structures found in the medulla of the thymus. They are the potent sources of cytokines, which has nothing but, I told you, they are the protein chemicals which are messengers that regulate the immune response. So this is something that was remaining to be told to you. So yes, these are the different immune organs, and as you know, they have their roles, they have their functions, and together with the cells and the intricate lymphatic tissue, they provide ultimate protection from antigens. So I hope you have understood. You can try practicing the drawing of the structures of these organs. If asked, a question comes for maybe 6 marks. So practice drawing this, okay? And yes, if you have questions, you are free to ask me. Okay, thank you for watching this video, and all the best.